thank you very much for working quite hard on the problem set. It made it easier to grade. I graded it very fast, so if you feel that I have been unfair about deducting points when you compare your answers with my answers, which are on Moodle, please don't hesitate to come talk to me. Announcement, we're going to have a biology colloquium a week from today, not today, but a week from today, on the coronavirus. So Lindsay, who is a virologist, a viral ecologist, will be talking about coronavirus, just in time for you to travel and bring the virus back to Irwin with you. Informative. Hopefully not. Let's just say hopefully not. But you know, travel is how this virus gets around. So a week from today, put it on your calendar and think about attending. That'll be upstairs in 300. So today we are going to talk about chemical senses. We can detect chemicals. We detect them with our sense of smell and our sense of taste. And so we'll do smell first. We will learn about the anatomy of the nose and the olfactory bulb, molecular mechanisms for how we transduce an odorant, presence of an odorant in the air around us, into an electrical signal that our brain can interpret. We'll learn about a map, sort of map of olfaction in the olfactory bulb. And then we'll talk about olfactory and gustatory taste cortices in the brain. So first smell, then taste. So we're gonna, as I speak, we're going to do a little game. I have 5 odorants. Please, what I'd like you to do is, I'm going to put each of the 5 in each of these 5 columns. And so the odorant should work their way back and then front and then back. Let's zigzag this way. So this one will go up and down and up and down and up. And this one will go up and then Brittany will decide whether it goes to the right or to the left. Same here. Same here. Same here. Okay? The idea, what I'd like you to do is, don't let it be too distracting, but smell the odorant and see if you can immediately place a memory. Just what does it remind you of? You know, it's like an ink blot test, a Rorschach. Just what does this odor say to you? And then try to use your brain to figure out what is this odor? What's the name of this odor? See if you can do those 2 things. Just jot it down on a piece of paper as it's moving around the room. So that's going to be happening while we talk about our ability to transduce these odors into signals that our brain interprets. So you know that you smell with your nose. At the roof of the nasal cavity is the olfactory epithelium. That's where the sense of smell is first transduced. And we get odorants from the air, which you are doing, and we get odorants from chewing food that releases volatile flavors that travel up to the nose and contribute to our sense of flavor. We have taste modalities in our mouth, but a lot of flavor is our ability to smell things in our nose when we chew and release flavor odorants into the nasal cavity through the nasal pharynx. So we're focusing on olfaction, but it actually is related to taste. At the roof of the nasal cavity is a specialized bone called, part of the skull, called the cribriform plate. And it's perforated. It's perforated because nerve axons from the olfactory epithelium. So this is the top, the roof of the nasal cavity. This is the bone at the roof of the nasal cavity. Here is the olfactory epithelium that interfaces between the bone and the air in the, or the mucus really, of the olfactory nasal cavity. And so this cribriform plate is perforated, and if you're interested, you can come and see in the skull the model, just looking at the specialization of the bone here at the cribriform plate. You can actually see some of the perforation techniques. I don't want to have too many things moving around the room, so we'll maybe pass that around at the break. So odorants in the nasal cavity bind to receptor molecules on the cilia of the odorant receptor cells. So the receptor molecules are on the cilia of the receptor cells. We'll talk about how that is transduced into an electrical signal that depolarizes these cells, triggers action potentials in their afferent axons, and makes a synapse in the olfactory bulb, which is on the other side of the cribriform plate. So, quick question. Are olfactory receptor cells short or long receptor cells? They're long. They have axons. We can detect thousands, some say millions, but that's debatable. There's actually some debate in the field about how many odorants we can detect. It's definitely in the thousands. It might be tens or hundreds of thousands. It could be more than a million. Debatable. Definitely in the thousands. Probably closer to 10 to 100,000. Here are 5, or 6 or 7, right? So a small smattering of the chemicals that we can detect by odor. So we can detect aldehydes that are usually fruity, nutty smells. We can detect thiols, which are skunky smells. We can detect rose, ginger, just a few, clove, the smell of clove. We can discriminate enantiomers. We can discriminate the R enantiomer of carbone and detect mint, and the S enantiomer of carbone and detect caraway, like rye bread. So very different odors triggered by very similar chemicals. So it's pretty interesting. I will not test you on this. I will test you on this. This is the mechanism by which all odorants are transduced into electrical information. So let's walk through it. You know this already. Most of it. What kind of receptor is this? GPCR. Odorant binds, conformational change of the G protein, GDP dissociates, GTP associates, beta gamma separates from alpha. In this case, it's the alpha, it's the GTP liganded alpha that activates adenylyl cyclase. You've been here before. Adenylyl cyclase hydrolyzes ATP into cyclic AMP. You've been here before. The G protein in the olfactory system is called G-olf, not golf. G-olf. But you might think, wow, G-olf must be very similar to one of the G proteins that you are very familiar with. Which one is it? GS. G-olf, like GS, activates adenylyl cyclase. It is not GS. but it is like GS. So everything you know about GS can be applied to GOLF. Yes?
So what's the difference?
There's some amino acid differences. I mean, they're just molecularly different, different enough that they don't call them the same thing. In this setting, so you know that GS can activate PK, or GS activates adenylyl cyclase, cyclic AMP activates PKA. There's the pathway you're accustomed to. In this pathway, we don't have to go to PKA. The cyclic AMP activates a cyclic AMP-gated cation channel. A cyclic AMP-activated ion channel. The ions are cations, and they flow in. You know what that does. That causes a depolarization of the cilia. Remember, we're in the cilia. Depolarization of the cilia. That depolarization migrates to the rest of the cell. Above a threshold, we will get action potentials. And the higher above the threshold and the longer above the threshold, the higher the action potential frequency. Action potential frequency, action potentials from the olfactory epithelium, where we were here. This is the olfactory epithelium. Action potentials, synapses in the glomeruli of the olfactory bulb. So the first synapse of the olfactory pathway is in the olfactory bulb. So at this level, olfaction is really simple. Some anatomy, some physiology, most of which you already know. Humans express something like 350 different olfactory receptors. So here's a cartoon of the GPCR. 1, 2, 3, 4, 5, 6, 7. This is the model with 7 transmembrane helices, with each of the amino acids in the receptor drawn as a circle, some of which are colored and some of which are not. So, and the color is blue are amino acids that are highly conserved from one receptor to another. So are black. Red and white are amino acids that are more variable. So you can see there's lots of variability in the different olfactory receptors, and we humans express something like 350 of them. Dogs express something like 1,000, rats express something like 1,500, because they are more olfactory animals. Humans express something like 12 million different receptor cells in our olfactory epithelium. A bloodhound expresses something like 4 billion of them, because it's a very olfactory animal. But the difference between the different receptors are, different amino acids. So one receptor will, different amino acids make different receptors. They all follow this pathway. So you're probably confused now. If we only express 350 receptors, and we can detect, let's say, 10,000 odorants, how do we do that? How do we detect 10,000 odorants with 350 receptors? Charlie?
I was just going to ask, is there a linear relationship between number of cells in the epithelium versus like olfactory ability? Like, are bloodhounds, whatever the multiple as, like,
I'm sure it's not linear.
much better.
They are better than us.
Yeah.
Are they, how many orders of magnitude are they? 1, 2, 3, almost, or over 4 orders of magnitude. Is that right? No. Anyway, yeah, 2 orders of magnitude. Are they 2 orders of magnitude better than us? Yeah, they might be. I'm sure it's not linear, but this does reflect the ability. I mean, a bloodhound can trace a really faint odor and follow it, you know, to its source. And we can't come close to that. So it might, I don't know the relationship, whether it's linear or hyperlinear or sublinear, I really don't know. All I do know is that bloodhounds are better at it than we are, and bloodhounds are better at it than a typical dog, and there seems to be some relationship with the number of receptors. I mean, number of receptor cells. So how do we use 350 receptors, molecules, to detect 10,000 odorants? It's called combinatorics, combinatorial activation. So let's just look at this. So here is octanoic acid, or maybe it's this one. I'm not even sure. One of these is octanoic acid, and here is receptor 1. Receptor 1 is highly activated by octanoic acid, but it's also activated by shorter chain carbons, heptanoic acid, and it's also activated by a bromo-octanoic acid. So this receptor is activated by a limited set of odorants. This odorant activates a set of receptors. The fact that we have this array allows us to discriminate. So for example, octanol and octanal, which are very similar to each other, we can discriminate because of the slightly different combination of receptors that are activated by those two chemicals. Octanol, the alcohol, octanal, what is that, aldehyde? Rusty on my chemistry. They're similar smells, orange, rose versus citrus. I mean, subtle differences because it's a subtle change in structure. Octanoic acid can smell sour, rancid. So very small changes in structure produce detectable changes in sensation that we can give words to. And the reason we can do that is that, in the word of Nobel Prize winning Linda Buck, each receptor recognizes multiple odorants, and I think she should have said and, each odorant is detected by a unique combination of receptors. It's the combination of receptors that gives us the ability to detect thousands of odorants with hundreds of receptor molecules. So that's a little tricky. It's not one-to-one. It's combinatorial. I hope that didn't spill.
Didn't spill.
Oh, thank you. They would hate me in this room forever. Please don't spill it. I mean, the smells are not terrible smells, but still. So the axons, let me go back to this. Each receptor type, so that's what this color coding is supposed to be. Each receptor type, let's just say this blue-purple one is receptor 1. Receptor 1 forms synapses in a specific glomerulus of the olfactory bulb. So we have 350 different types of glomeruli in the olfactory bulb, where the synapses from receptor 1 form. Remember, receptor 1 is activated by a combination of odorants, and each odorant activates a combination of receptors. So we have different levels of activation of different clusters of cells, clusters of synapses in the olfactory bulb. So synaptic processing of the odorant information in the olfactory bulb, and then some of these glomeruli, or different patterns of activation of the secondary cell, depending on the nature of the input to the olfactory bulb. And these axons from the olfactory bulb go through what nerve? you know what it is. The olfactory nerve. Right? Cranial nerve 1. Olfactory. Cranial nerve 1 then innervates a specific part of the brain called the olfactory cortex. Now, so let's do a little bit of review. Here is the post-central gyrus, where the somatosensory cortex is, the primary somatosensory cortex. Here's the anterior, posterior occipital lobe, where the primary visual cortex is. Here is the the superior temporal lobe, where the primary auditory cortex is. So this slide has some information of things that you know. It's opening up the lateral sulcus to see structures sort of underneath it. The olfactory cortex is in the medial temporal lobe. You've heard those words before. What else have I talked about that's in the medial temporal lobe? The hippocampus. What is the hippocampus important for? Memory. The olfactory cortex is physically located near the hippocampus and the amygdala and a lot of the parts of the brain that are associated with motion, memory. And that's why I asked you, take a smell and see if you can trigger a memory or a feeling or what it reminds you of before you try to use the rest of your brain to identify the name of it. It doesn't always work. The olfactory cortex is in the medial temporal lobe, and for those of you who are experts, this is called the uncus. I won't test you on that word, it's not on the key concepts list. By the way, if you have lost your key concepts list in preparation for the quiz, another break, quiz on Sunday, we'll cover material up to and not including today. Mostly anatomy, up to through Tuesday. I will not, I'll just be honest with you, I will not go retrograde and ask you about muscles on this quiz. So, back to where we were. Olfactory cortex in the medial temporal lobe in a part of the brain called the uncus, near the amygdala, near the parahippocampal gyrus, near the hippocampus. We're going to see in a few minutes, we're going to get to taste, and the taste cortex is actually very close to the smell cortex, the olfactory cortex. But it's on the other side of the lateral fissure in an area called the insular cortex and the frontal lobe. Anyway. So as I have asked you to learn about where the primary somatosensory, visual, and auditory cortices are, I'm going to ask you to know where the primary olfactory cortex is. We haven't talked about motor cortex yet, but we're going to talk about that starting next week. So information from the olfactory bulb goes to the primary olfactory cortex, but then it goes everywhere, right? Just like everything else. This is supposed to be this is an fMRI showing sort of a, right? So here's the corpus callosum. So we're seeing part of a horizontal section and part of a midline sagittal section in this image, and you can see different parts of the brain activating in response to the odor. So now, before we get into taste, let me get my answer key. Okay. So, odorant 1. Let's just play this game a little bit. What did odorant 1 remind you of, Dorothy? A what cleaner? Oh, bathtub cleaner or a cleanser.
Yeah, common.
Yeah, okay. What else? Anybody else? Yes. Sure. So,
Eucalyptus.
Eucalyptus, okay.
Pine trees.
Pine. Pine. So these are the names of it. Did any of, so the name of it is pine. So it's a pine scent. Did anyone, instead of, before you got to pine, did you remember, oh, a walk in the woods, or something like that? That's what I'm getting at. So it's hard to do in a classroom setting. Austin, did you have a question, or did you want to say something about pine? Or?
I wrote down forest.
Forest. Okay, so forest. So it's like that's not a smell. That's not a name of an odorant. That's a memory. That's a place. Okay, let's do number 2. First, before we name it, did it remind you of anybody? Did it remind you of some event? Did it remind you of some place?
I felt like I was taking a walk in a library.
Library. Okay. That wouldn't have occurred to me, but I know what they are, so I'm messed up. I mean, in terms of this. Anybody else? Yeah, Dorothy. We're talking about number 2. Of what? Flowers. Okay. What's its name? Lavender. Anybody else want to think of a different flower? That's not the name of the odorant. Jasmine? Rose? How many people have smelled rose? It's rose oil. But this is really interesting. It's hard. It's harder than you would think. You smelled something flowery, but it was hard to come up with the actual name of it. It was rose. It's rose oil. Rose water. Rose water. And in the past, some people, so as people get older, there's less and less of rose perfume. But in the old days, you put this in front of people and they say, oh, that's my grandmother, or that's my mother, or that's Aunt Frances, or whatever, because of the perfumes that these people used to wear. It's not happening as much anymore. Okay, number 3. Any memories or events associated with odorant number 3?
The holiday.
The holidays. You mean like Christmas?
Yeah.
Rebecca.
Walking into Joanne Fabrics.
Joanne Fabrics.
I'm gonna say Christmas too.
Christmas. What's its name? Clove. This is clove oil. So it smells like Christmas because, you know, they, mulled wine has a lot of clove in it, or there's a thing that you can do putting cloves in oranges that make a very fragrant odor, smell in your house. In the past, I'll just be honest with you, in the past there have been people who say, pain, this reminds me of pain. Not because of Christmas, but because dentists use clove oil as an anesthetic when, in the case of tooth pain. So before there is a root canal done, they often pack the tooth or the cavity with clove oil as an anesthetic. And so that smell, for some people, reminds them of intense pain, because tooth pain is really intense. So that's interesting. Depends on your experience. Christmas, you know, the happy smell of Christmas, or the intense smell of pain. Okay, number 4. Anybody got a memory, or just shout out something? Memory or a name? Lemon. Lemon. Yeah, this is lemon, right? So some people have in the past shouted out cleaner, you know, because they often put lemon scents in cleaning fluids. Pine's, well, pine's always pine, but you know, dishwashing liquids and stuff are often lemon scented. And number 5, peppermint. That one's pretty easy because you put that in your mouth all the time. So these, just 5 different odorants. Some of them are easy, and you know they immediately trigger a word. Some of them are less easy. They trigger something, foresty, flowery, but it's hard to come up with the actual odorant. And so we're not very good at making this connection, although we're pretty good at making memories associated with odors. Yeah.
If he was sick or like he noses uncongested, how would that
Okay.
This, or like, looking for-
Right, so if we're making a lot of mucus, the mucus makes a thick layer on the olfactory cilia and prevents odors from getting through the mucus, and so there's a barrier to the odorant getting to the cilia. If our nose is congested, nose is full of mucus, it's just prevents the odorant from binding to the cilia. Other questions? Okay, let's move on to taste. So remember, we're not talking about flavor, because flavor is mostly smell. We're talking about taste, what the detection of tastes on your tongue and the back of your throat. And we have only 5, well, some say 6, but 5 taste modalities. We can detect sweet, salt, umami, basically the taste of meat, bitter, and sour. Those are the 5 taste modalities. Umami is the Japanese word for delicious. And it has its own sensory modality. Some say that we have a spicy hot flavor modality. It gets into culture now, right? In Western culture, these are the 5 modalities. In some other cultures, spicy hot is a taste modality. It's part of the culture. And, but it doesn't have a taste, well, you know what spicy hot binds to. It binds to the pain receptor. So in Western culture, this is not a taste modality, it's a pain modality. But in other cultures, no, that's not pain, that's taste. So now we're getting into some cultural neuroscience here, and I won't test you on it. Question? Did I see a hand? Yeah.
What molecule binds to spicy? Capsaicin.
This is capsaicin. Capsaicin receptor. We have capsaicin receptors in our mouth, and so we certainly taste pain or spice, depending on your perspective. We have capsaicin receptors. There's also there's a slightly different receptor that detects the heat in wasabi. It's not exactly the same receptor. It's a highly related receptor. Now you'd say, well, wasabi's not pain, that's taste. So that lends some argument that no, it's not 5, it's 6. And the 6th being sort of spicy hot, wasabi flavor, capsaicin flavor. I'm open-minded about this. The textbooks will say 5, because the textbooks were written by Europeans, Anglo-Americans or something. Yeah, Charlie.
Does anyone know then, is the Scoville scale
Yeah.
is it like a scale of affinity for different spice molecules too?
The Scoville scale is the amount of capsaicin in a certain amount of food.
Okay, so it's just amount. And
It's
not-
just a concentration of capsaicin is what it comes down to.
Okay.
So peppers with 1,000 points on the Scoville scale are super hot. And, you know, green peppers are not super hot, but they have they're 1 or something on the Scoville scale. Scoville, some scientist who made a scale of hotness for various peppers. There are ghost peppers that have 10,000 or something on the Scoville scale. They hurt. They hurt a lot. I could tell you a story about my son, who, when he was in 3rd grade, accidentally broke open some habanero peppers. He and a girl in the backseat of our car had got their hands on habanero peppers, and they didn't know what they were. They're cute little peppers. And they opened them up, and they got the juice all over their fingers, and then they got them all over their face, and soon they were screaming. And we had to take them home to try to clean off and put aloe on their skin. I mean, they had chemical burns it looked like chemical burns anyway, from the inadvertent spreading of capsaicin from habanero peppers. He still doesn't like spicy food very much. So taste is detected by taste receptors, and this is a taste bud. It's not glossopharyngeal, so I'm sorry about that. That shouldn't have been linked to this image. This is a taste bud on your tongue. We also have taste receptors, not in buds, but expressed on the back of our throat. So we taste with our tongue and with the back of our throat. This is a taste bud. The receptor cells are, there's several different types of receptor cells in each taste bud. The receptor cells make synapses in the taste bud with afferent neurons. So here's my classic short question. What type of receptor cells are taste receptor cells? Short, I gave you the answer. They're short. The receptor makes a synapse right there in the taste bud, and then the action potentials are in the secondary neuron that go to the taste cortex. Well, actually, they go to the thalamus first, and then to the taste cortex. Oh, interesting fact. Every sensory modality that we've been talking about, somatosensory, visual, auditory, olfactory, taste, goes to the thalamus and then to the cortex, except for olfaction. There is no olfactory processing in the thalamus. I won't test you on this either, but it's interesting. There's a direct connection between the olfactory bulb and the cortex, whereas in all the other modalities, there's thalamic processing. Why? I don't know. Anyway, taste bud. So here is a cartoon of the taste receptors in the back of the throat, in the back of the tongue, in the front of the tongue. You may have heard that that we taste sweet with the front and bitter with the back, and that's not true. The taste buds at the front of the tongue have a sensitivity to the sweet, but they also detect all the other modalities, even bitter. The taste buds in the back of the tongue, the posterior part of the tongue, have a higher expression of bitter taste, but they also express sweet receptors, so they detect sweet as well. So the thing that you've heard, sweet in the front, bitter in the back, is a gross exaggeration. It's not completely wrong, it's just an exaggeration. So the taste buds at the front have a certain name called fungiform, shaped like mushrooms, I guess. The taste buds in the back have another name called circumvallate. These are not on your key concepts list. I will not ask you to memorize those names, just that you can show off to your friends. The taste buds in the front, the fungiform. Has anyone taken biodiv? How do they say that part of that word? I know how you say fungus, but how do they say that word? Fungi. Fungiform. Okay. You have to remember this. Fungiform. The fungiform taste buds express different receptor cells that express different receptor molecules. And the same with the circumvallate, and the same with the taste receptors in the back of the throat. We have 3 cranial nerves that carry taste information to the brain. The vagus nerve, the glossopharyngeal nerve, and the facial nerve. All 3 of these cranial nerves carry taste information. So this is where the sort of, the olfactory nerve carries olfactory information, and the optic nerve carries optic information, and the vestibulocochlear carries auditory and balance information. It's pretty one-to-one, except for taste. Taste, it breaks down. We have 3 different cranial nerves. Vagus, mostly innervating the back of the throat. Glossopharyngeal, mostly innervating the back of the tongue. And facial, mostly innervating the front of the tongue, carrying taste information. Will I ask you to know that? Yeah, I will ask you to know. But I'll probably say, name name the cranial nerves that carry taste information, and I would accept 2 out of 3 or something like that. Dolly?
I guess I think water is like taste.
I'm not sure what this is getting at. So let's just ignore it. Taste, unlike olfaction, is complicated. Brittany, is it okay if I go over here for change? So remember, we've got 5 different modalities, and we have 3 different mechanisms. Okay, so the simplest one, the one that uses material that you already know, is sweet, bitter, and umami. Those 3 taste modalities all activate a GPCR. The whole GPCR story that you know, should know. The GTP liganded alpha and the GTP liganded alpha and the liberated beta gamma both activate phospholipase C, a type of phospholipase C called phospholipase C beta 2. I won't ask you about the beta 2. What type of G protein is this most similar to? GQ. In fact, it is GQ. So the GQ story, phospholipase C, diacylglycerol and IP3, the IP3 receptor activated in the endoplasmic reticulum, calcium release, and now the new stuff. We're not talking about PKC here. We talk about calcium activated channels. Calcium activated sodium channels. And so influx of sodium, depolarization of the taste receptor cell. So this is, and let's not worry about this annex 2. Just sort of cross that out. Don't worry about that. But you do want to worry about this pathway. Calcium, not activating PKC, activating a calcium-activated cation channel. Sodium channel. Depolarization, neurotransmitter release, taste. Sweet, bitter, or umami. That one's easy. Salt is super easy. There are salt channels, sodium channels. These are not voltage gated. These are sort of constitutively activated salt sodium channels in the taste receptor membrane. And when there's more sodium influx, depolarization, neurotransmitter release, taste of salt. So that one's super easy. All you have to know is a sodium channel that is constitutively expressed and constitutively open, salt is sodium. The tricky one is sour. So with sour is acid, it's the taste of acid. All of the acids are, in acid-base combinations. You've learned this in Gen Chem. Weak acid, weak acid what's the word in chemistry? Wait. In equilibrium with its conjugate base, I think that's the way they say it in chemistry. So the neutral form of the acid can go through the membrane. The protons can go through proton channels, and there's depolarization right there. And these protons that either got there from diffusion through the membrane or proton movement through protons channels blocks potassium channels. A block of potassium channels, going all the way back to the beginning of the semester, a block of resting potassium channels will allow depolarization. And we don't, so basically, this is this, you could cross out, everything else everything else is the same, or close enough. So you can just cross out the rest of these cartoons and just focus on this one. I probably should have done that for you, you can do that too. Okay, sour, acid influx, acid, block of a K-channel, depolarization, neurotransmitter release from the receptor cell. Okay, this is how we transduce tastes into electrical information. So, and just remember, this is a short receptor cell. So depolarization, calcium, we don't have to get involved in the whole thing. Depolarization, calcium influx, calcium, you know what this binds to, synaptotagmin, fusion of neurotransmitters, vesicles, neurotransmitter release, activation of a excitatory receptor on the afferent neuron, and action above threshold, action potentials, action potential frequency going to the thalamus, going to the taste cortex. There's some uncertainty about the nature of the neurotransmitter here, but we know that there's evidence for serotonin, norepinephrine, and ATP as being the neurotransmitter. I won't test you on that. But I would feel compelled to test you on the idea of a short receptor cell, depolarization, calcium influx through voltage-gated calcium channels, neurotransmitter release, and the details that I'm leaving out. Sophie?
So the inhibition by the protons creates a depolarization? Or
Exactly. The
Yeah.
inhibition by the protons blocks the resting potassium channels that were holding the membrane polarized.
oh,
Remember, potassium efflux is what causes polarization. Block of potassium efflux allows depolarization. Yeah.
So every, different receptors causes depolarization and more calcium influx?
Right.
How do you distinguish
Oh,
between-
different receptors expressed on different cells. So different receptors. So this receptor cell expresses umami receptor. And this receptor cell expresses sweet receptors. And this receptor cell expresses sour receptors. So different receptor cells express different receptor molecules that each activate a different pathway. But remember, this receptor cell, what did I say, umami, is connected to a set of afferent neurons that is carrying action potentials to your brain, and your brain says, delicious, umami. This receptor cell is connected to a different set of neurons, different set of axons that go to the brain and say, ooh, sour. Did that answer your question? So let's take a break. Okay, one more question and then we'll take a break.
In each taste, is there like level of taste? Just
Sure.
like,
So you eat food and you have some bitter and some sweet and some salty, and that combination is what we produce as flavor. I can activate one, so I can give you salt and we would activate salt only. That wouldn't taste very good.
So it's just like salty is salty. It's not like less salty or more salty.
Oh yeah, sure, the more salt, we
And
certainly,
the receptor should be different.
we have, yeah, we can detect more or less salt. You know that you can detect really salty food or not so salty food. And so, salty is active, if we're at, there's a concentration dependence in the amount of influx of the sodium based on the concentration of sodium. And so that amount of influx, activating more receptors, more depolarization, more action potentials tells us, oh, more salty. So let's take a break. And I've got a boatload of clickers that we can use to sort of summarize, because where we are right now is we've done the sensory inputs. Somatosensory, touch, pressure, pain, vibration, visual, auditory, taste, smell. Am I skipping one? No, I think. So these are our sensory inputs. Next week, we're gonna start to talk about outputs, movement. You know, at a really basic level, we're input-output machines, right? We get input and we then produce an output. Of course, we're human, and so this doesn't get into memory or the emotion associated with inputs and outputs, but we're gonna stay simple and think of, we've talked about the inputs. Next week, we'll talk about the outputs, the motor system. You know some of it because you learned about muscle in self-is. I'm gonna build on that next week. So we're at a transition, kinda. We're a bunch of clickers, some of which we'll get to, we'll do some of them today and have a conversation about this information. And what we don't get through is up on Moodle for you to review for the, well, in some cases for the quiz, and then later for the exam. The exam is the Thursday after break. So don't get surprised when you come back from break that we're pretty much right on the exam. Okay, let's take a break. Let's start with the first clicker. Which of the following sensory systems uses GPCRs? Visual and olfactory. Auditory system uses stretch-activated channels. Somatosensory system uses either stretch-activated channels or pain chemical activated channels, like ATP or acid. Okay, now we're going to do a little GPCR review. The GTP-bound alpha subunit of transducin, what does it do? Remember which one is transducin and what it does.
Sorry.
This is transducin. Transducin is in the visual system. The GTP-bound alpha subunit of transducin is what activates a phosphodiesterase in the rods and cones that breaks down cyclic GMP and allows the closing of those cyclic GMP gated channels. Questions? I want to give you the opportunity to say, wait a second, I thought da-da-da-da. Okay? The GTP-bound alpha subunit of G-OLF activates adenylyl cyclase. The channels are activated by cyclic AMP. In photoreceptor cells, cyclic GMP, what does it do? Back to photoreceptor cells. It activates cation channels. In the dark, remember the story in the dark, the guanylyl cyclase is making cyclic G. Cyclic G is opening channels. The cells are, the resting potential of these retinal cells are polarized, relatively depolarized. There's lots of neurotransmitter release. When light hits the receptor and activates transducin, the phosphodiesterase cuts cyclic G, allowing those channels to close. So this physiology is something to learn. These clickers right now are focusing on physiology. which will be on the exam, but won't be on the quiz. Just want to be clear about that. Okay, in auditory hair cells, stretch of the tip link. Activates cation channels. In olfactory cells, cyclic AMP activates cation channels. Okay, I'm going to skip a few because I want to get to some. Let's do this. In photoreceptor cells, auditory hair cells, and olfactory cells, activation of cation channels, what does that do? Give you a hint, there's two good answers on this one. A and C. Depolarizes and then leads to opening of voltage-gated calcium channels. Okay, now we're going to get into some anatomy. So this could be on the quiz or on the exam. In which of these sensory systems is the receptor cell a long receptor? C and D. Visual system, the rods and cones are short. Auditory system, the hair cells are short. Olfactory system, we just did this. The olfactory receptors with their cilia have axons, they're long. And in the somatosensory system, all of the cells that detect touch, pressure, pain, vibration have axons that go to the spinal cord. A difference between a long receptor cell and a short receptor cell is good, you're all getting it. That's the difference. That's the key difference. Okay, so these are more physiology. I'm going to skip them and see, folks. Okay, I screwed this one up. This is getting into some anatomy. So let's do this. Knowing where the first synapse is part of knowing the pathway. So the first synapse of the pain pathway is in the dorsal horn gray matter of the spinal cord. That's something you learned and something I could quiz you on. The first synapse of the touch pathway. In the medulla. If you wrote thalamus, you're not far off, but it's not quite right. Okay, the first synapse of the visual pathway. It's in the retina. The first synapse is the synapse between the photoreceptor cell and the bipolar cell, right there in the retina. The first synapse of the auditory pathway. Is in the corti, right there in the cochlea, between the auditory hair cell and the afferent neuron. Questions about this anatomy? First synapse of the olfactory pathway. It's in the olfactory bulb. In the first synapse of the taste pathway. Is in the taste bud. Yeah.
Is there something called
Taste cortex? Yeah. Let's see, so, I'm gonna scroll back, rather than, let me just. The taste cortex is, it's in this area of the brain called the insula and the frontal cortex. It's near, if we were to put this back, it's near the olfactory cortex. But the olfactory cortex is in the medial temporal lobe, and the taste cortex is in the insula and the frontal lobe. Joey, do you have a question? Other questions? Okay, let's, so there's some more physiology. Just in the interest of time, we're going to skip those. These are challenging, so let's do these. Oh, I don't know how we can do these as a clipboard, so. Okay, so we can't do this. So I'll let you do these at home. Homework for the test. We could do this one as a kicker. So, pain sensation is associated with which of these? Pain sensation is really most associated with, so there are, this is one of the ways that we detect pain, is by detecting the acid associated with tissue damage. There are other ways of detecting pain. The capsaicin receptor, ATP receptors. So this is not a comprehensive answer for this, but it is one of the ways that we detect pain. Okay? Visual system. Which of these is most associated with the visual system? Right, you're getting cyclic G, David Catlin chains. The auditory system. Stretch activated channels. Olfactory system. Which of these, if any, is associated with the olfactory? None of those. It's cyclic AMP gated cation chains. taste. Which of these is associated with one of the taste modalities? I'll give you a hint. There's one best answer. We have acid blocked potassium channels. We have salt permeable sodium channels. We don't really have acid activated sodium channels. We have acid activated sodium channels in the pain submodality of the somatosensory cortex, somatosensory system. Okay, so we could play the game. Name things. You can do this at home, right? What's the name of the sulcus that's associated with the visual cortex? Anybody got it? Calcarine sulcus. Can't really see it from the outside, so this is kind of imaginary, but you can see it from the midline sagittal section. I finally learned the name of the two gyri above and below the calcarine sulcus, which, if you're interested, I can tell you, but I'm not going to burden you with that information because I didn't ask you to learn it. What is the name of this structure where the auditory cortex is?
Temporal. The superior temporal.
Superior temporal gyrus. What's the name of this sulcus that separates the motor cortex, which we'll talk about next week, from the somatosensory cortex? Central sulcus. So this is anatomy. I could ask you questions about this. I could ask you to draw a schematic of a cross-section of a spinal cord and label it with things like this. So we're not going to do this one here because, and the answer is on the next slide, which I'm not going to show you, but you can do this as homework and then check your answer against the next slide. So your key to the quiz is knowing that it's anatomy, the anatomy we've covered in the sensory systems, your coloring book, PowerPoint slides, your notes, the key concepts list. Focusing on anatomy is what you want for the quiz. And then that will also come back. I'll do it again on the exam. That's not till after the day. Are there any questions about leading up to the quiz, which is due on Sunday? I'm okay with the do we need to know questions? Yeah. Do we need to know the mapping in like the somatosensory cortex? Yeah. You do need to know the sensory maps. Okay, well, I'll see some of you tonight. I'm available today, a little bit today, sometime tomorrow if you have questions. Don't forget to do the quiz. The desk closes. What time does the desk close on Sunday? Midnight? Okay, so I just want to make sure that's still true. Okay. All right. Have a good afternoon or rest of your day.